and welcome back to a proper edition of the Ask podcast today. I think this is episode 8. I'm starting to lose count now. But we hope you will be attaining some secret knowledge with us and our fantastic guests and panellists today, who I'll introduce in a moment. Just to remind you, I am Mrs. Jenny Summers, Deputy Head of English and Grammar. Oh, that's good. English and Grammar.
I see the subject as mainly grammar. We're going to change that job title.
That's interesting.
We're not editing that out. That's staying in. That's staying right in.
Okay, fine. I'm Deputy Head of English and Drama at John Hampden Grammar School. Last time I checked, and hopefully I'm not going to be fired after this podcast. And I'm really excited for this episode today because the subject is not in my area, my comfort zone necessarily. So I'm going to learn lots, I am sure. And this is my lovely colleague.
I am Mark Till. I am the Head of English and Grammar. I oversee all the grammar. This is going to be a great one. We've been trying for a number of weeks, haven't we, to have a science colleague. And we have an actual science. We've trapped one in the room, like when they do those experiments where they try and trap an electron. I feel like tracking down scientists is that difficult. And we've finally managed to get one from the wild. And this is going to be fantastic. And we'll do another clue for clue hunters. Ooh. Another big prize. We've only had one clue, and there will be another clue, but it will be dropped in at some random point.
Absolutely. Not right at the end.
Not right at the end if we forget again, possibly.
Good. And we've got some lovely brand new panellists this week as well, including Mr. Miles Kingman, whose birthday it is today. Happy birthday, sir. Tell us a bit about yourself.
Thank you very much. So I'm an old boy from John Hampden, currently doing the classroom assistance role. It's a bit of a COVID catch-up. And then next year I'm doing my maths PGCE.
Very exciting. Joining the ranks of the teachers.
Joining the dark side, indeed.
Oh, the dark side.
Depending on your outlook.
Well, yes, absolutely. And we also have Suresh, who I've dragged in from my form S3, the best form, I'm sure you will all agree. Suresh, tell us about yourself.
Hi, I'm Triyesh, A-level student taking maths, further maths, physics and computer science. So I do a bit of science, but it's not chemistry. So.
He's getting his disclaimers in early. And finally, we also have Andres.
Thank you, Mr. Summers. I study chemistry, biology and maths, so hopefully that will give me some mild understanding of the subject that we're going to talk about today.
I like the fact we're all couching this. We're getting in our excuses early. We're terrified because we've just... The intellect that is our guest today, our Dr. Anthony Bullard, Head of Science, and he's formerly even been Head of Maths too. He could be Head of anything if he fancied it. You know, he's a very esteemed guest. And he's got some really interesting things to talk to us about today, about being a scientist, about being a research scientist, about training to become a scientist, and perhaps we might even pop some literature in at the end if we have time. You never know. So why that topic, sir? Why did this topic tickle your fancy, first of all?
Well, you asked me to do something, and I was trying to think of something that might be interesting to both parents and pupils alike. And we do a lot of science here, and in terms of a lot of pupils who do science and go on to do scientific careers and thereafter. But what we don't really do is what it is to be a professional research scientist. And it isn't quite what the glamorous view that most people have of it. So I thought, particularly as a lot of the lower sixth are used to me ranting at them about some of the things that I don't like about the curriculum and about the thought process and training to become a scientist, I thought it might be kind of interesting to get a glimpse for pupils and even parents about what it is day to day like being a scientist. What are the skill sets you need? What are the thought processes you go through? And what are the deep frustrations and unglamorous bits that you have to go through to be a research scientist?
Excellent. I was just wondering, before we get into all of that, what drew you into a life of science in the first place?
As a sixth form tutor, this is going to be really bad because I keep saying, you don't need to know what you're doing by the age of 16. Don't worry, it'll come to you. It's really normal. I hate to say it, but I used to watch Horizon, which was a documentary series back in the day in BBC Two. And I used to see medical research, and I think, I'd like to do that. So sadly, I've always wanted to do research science, and particularly related to the medical field. Had a vocation for it.
And what was your story then? How did you get from that? What did you go into first? Give us a sense of your journey. You don't have to be too personal, but a sense of
I went to Surrey University to do molecular biology, and I chose molecular biology because I kept on asking the question why. And if you're in biology, and biology is my field, You say, why does an animal behave like it does? Well, then you get down to the cell level. Why does a cell behave like it does? We'll get into subcellular, and then you get down to the genetic code and the interactions, and you keep on asking, but why, but why, but why? And that leads you to molecular biology, which is, the chemical interactions and machinery of what's happening within the cell. And I chose Surrey because I had an industrial placement. I mentioned this to a lot of my tutees. So I felt my sister had gone there, and she'd had a really great experience. And I felt that even, whenever era you look at, having a year's work in industry before you graduate actually on your CV puts you at a much better advantage compared to people who leave. Plus the fact you actually earn genuine money. It's not a huge amount of money, but it feels like a lot of money when you're that student. So I did that, and then I was offered some jobs that are coming out. I didn't have to look too hard. I was offered some jobs that are coming out because of my year in industry. And I worked in the pharmaceutical sector for a short while thereafter, but it was pretty unsatisfying. I didn't enjoy it particularly very much because I felt I was locked into a field that wasn't really mine. I was ploughing a furrow that wasn't one that I wanted to go in. So I did a master's in molecular medicine at UCL, which was the best year in education I ever had. It was just fascinating. They were bringing in experts from all over the world because the reach of UCL, and you were literally having people lecture to you that wrote all the papers, and you could question them. And I was that annoying person that kept on asking questions, and everyone else wanted to go to lunch because I was just having such a great time. And I got so much out of it, how to write scientifically. No one had taught me how to write scientifically, and they don't in the education sector. And I really struggled in essay writing, but thereafter I didn't. And then from that, the last term is meant to be doing a dissertation, and they pop you into a field at UCL, and I was meant to be doing about genetic mutations in deafness. And I'd agreed it all and discussed it with the lecturer and got on really well with her. And then this job came up in the field of cardiac research. And they said, well, you could either be earning nothing doing your deafness, or you could be earning money and doing your research with this cardiac group. And I thought it was quite interesting, popped along, and then ended up doing my research dissertation with them, and that led into a PhD. So that's my story.
Oh, great story.
Now our panellists get to be those annoying people who ask those questions.
I think my first question is why molecular biology? Why not just biology? Because when I was going to choose my degree, I was looking at physics, and I was wanting particle physics and quantum physics, but my teachers were pushing me away from a more specific point. Do you think that may have held you back? Do you regret doing something more specific so early?
Not at all, because I went to Surrey, and I think it's if you look at the rankings for Surrey nearly always comes out top for employability at the end. Okay. That's partly the industrial placement, but partly the flexibility of the course, which I think lots of other universities now copy, which is the modular system. So you can do, and the first year is pretty biochemistry, which is a pretty general course. So 6 of the modules are biochemistry, and all the nutritionists do it, all the microbiologists do it. Lots of different courses do those 6 modules. So if you want to, you can just choose different modules and plot a different pathway. But why I chose molecular biology, and what I tell my pupils in my tutees is, I just literally read the course code, literally read the prospectus, and you feel, you've got an intellectual interaction with your course, and you think, yeah, that makes a lot of sense, I got a career out of that. But there's also an emotional interaction with it, where you feel your blood pressure slightly rising when you read those course codes. And I think that's when your head and heart say the same thing, that's when you go for it, I think. So, it was partly emotional, partly intellectual, and also the question, but why, and that leads you to molecular biology.
And when they marry up, that's kind of just the right decision at that point, isn't it?
I've never regretted it, and particularly at Surrey, where I could do all sorts of things in my final year. I did a module in forensic toxicology, for example.
Oh, nice.
And all sorts of things. Pharmacokinetics and all sorts of interesting things, which I wasn't expecting to do to begin with.
No.
I couldn't even tell you what those things are.
I'm going to use I won't use jargon. Pharmacokinetics is the distribution, absorption, distribution, metabolism, and excretion of drugs through the body.
Okay. I just
And
about got that.
It's particularly relevant to the pharmaceutical industry. What do drugs do? How do you take drugs in? How do they interact with your body? And how do you excrete them?
Teachers and coffee, that's all I'm thinking now.
It's interesting. My head went to the PSD lesson I've just had, which was all about whether we should legalise cannabis or not, which partly talked about the
metabolism
impacts of that on the body and so on. So, very interesting.
You can't tell because this is a recording, but they're all looking at me. Well, I'm not so sure. Let's start with a general question. What annoys you most about current reporting of science in the media?
Yeah, good question.
Do you know what frustrates me the most? We live in this world, and we talk a lot about it in my tutor group, in this world where we all live in our own bubbles. and everyone is their own echo chamber. And it's kind of the way the population's gone, where it used to be the Guardian readers would have a certain view and the Times readers, and now it's just certain people follow certain people on Twitter. What frustrates me the most is when scientists do it, and they should know better, because they should follow the facts, but they choose facts to suit their purpose, and then they use the sort of the cover-up type of scientist. Yeah, you are a scientist. You should be choosing facts across the range and getting a broader view. And I find that frustrating when they don't. I don't find it frustrating when journalists do it, because that's their job, really. They peddle a particular view and they follow it, and that's their role. I find it frustrating when scientists do it as well.
Going back to one of the things you mentioned earlier, do you think there's limited freedom in the industry? Say you were doing your dissertation on deafness, but was offered a job in a course you say didn't want to do. Do you think you're being forced to do it?
Oh, there's a couple of things here, I think. If you look at the world of research, you can. I mean, funding is hugely difficult in research. You can have the best idea in the world. It leads to a Nobel Prize, but if no one will pay for you to do it, you can't do it. So funding is really, really difficult. And actually, the successful professors are those who bring in the money. And unfortunately, my professor, where I was very good at bringing in money, so we never had a problem getting the funding that we wanted. So one source of funding is pharmaceutical industry. But of course, the pharmaceutical industry want you to go down a particular direction. So that's fine, but so you have a bit of limited freedom if you follow the pharmaceutical industry funding. That was my experience with the colleagues that I had who attracted certain funding. And of course, they've got their research, which they want to patent, so you can't necessarily publish it. So that's quite tricky. Whereas working when I was doing my research in a university at UCL, you have almost total freedom. I could go to my professor, like, I'd follow this line, please, and he'd fund it, but I'd have to justify it. Say, look, this is the reasoning. Looking at these papers, I reckon this might be the direction I want to go down. And by and large, if I had a sound reason, we'd follow it. So there's a lot more freedom in the university sector. Is that the question you were asking?
Yeah.
It is the question.
That's always helpful.
Yeah.
Back to you, Sally.
Maybe going back towards the education part, you talked about being very frustrated with kind of what you teach now, and maybe how that might limit you in terms of going into this kind of scientific job. I think our ideas kind of marry up. Of my feedback from my A-levels were the idea of science being part of a wider scientific community was almost lost on me. The idea of reading journals and peer review, and that idea of a wider sort of working group was lost on me. It was very much focused on learning core content, which is part of it, but that actual, maybe the softer skills behind that were almost lost.
Yeah, I mean, there's that. I mean, I'm not too critical of the courses. I mean, they teach important stuff, the A-level courses, chemistry, biology, and physics. I think some of the frustration we find as teachers, and the things that I rant about, are what's been taken away is the concept of cross-disciplinary research. So when you go into research, you've really got to look at the world around you with a really broad view. If you're looking at it from one particular angle, you're really cutting out lots of ways of solving a problem. So what's happened with the A-level courses, so I take chemistry, which is the central science. That's how it's branded, it's the central science. And there used to be little bits of physics in there, crossover, you can see a little bit of physics. So you can see the interaction between physics and chemistry, and you have the mathematical basis of chemistry. And then also you can see how the organic chemistry, the biological end, leads into, say, the structure of hemoglobin and why blood is the colour that it is and that kind of stuff. All been taken away. Now, I understand why, because they want to not have somebody get double jeopardy where they learn one thing and they get double reward for it on two different A-levels. But it's taken away the concept that there's lots of different ways to look at things, and actually science is just the way that we look at the world around us, and actually subdividing into chemistry, biology, physics, and whatever other science you want to do is a way of us being able to compartmentalize it so we can chunk it down so it's actually manageable. But we should be, once you get to research level, you should be able to look at it with a really broad spectrum. And by having been trained in this really narrow thought process, it doesn't help when you get to the world of research. We drop off at the end, and there's no help, honestly. There's no help. There's no manual. There's no scheme of work. There's no method to teach you anything. And if you're looking at things from your narrow perspective that you've been trained in, I think it makes it harder. So that's the frustration I have.
Thank you for feeding in so nicely to the literal theme underlying our podcast, which is about fostering cross-disciplinary and cross-subject discussions. So that's very appropriate, I think. Should we? Because we've dotted about in some very interesting directions, and we've steered away slightly from the life of what's the word that I'm looking for? Researcher. That's the word. It's not that I'm an English teacher and I need to know words or anything. It's absolutely fine. Do you want to tell us about maybe a typical day in the life?
I think it works in weeks.
Okay.
I think if we give you a typical week.
Go for it.
As mentioned to Miles earlier, there's no manual for anything. All you've got are research papers where you look at methods and you look at the methods that other research groups are doing around the world, and you try and replicate them or adapt them and everything else like this. So really, you've got to manage yourself. Obviously, you've got the support of senior colleagues and your professors and postdoctoral scientists that will help you along. But really, you're in your own world. So a typical week will be you spend time reading papers. So the first thing I did when I sat down and started my PhD was I had this big folder of papers, literally the bang as it hits your desk. Right, read that lot and then we'll talk, is what the professor said. And then, so you're basically trying to plan out your week in terms of the research and the practicals that you would like to do. And one of the difficulties is that the practical lengths may not necessarily match your working day. So, for example, one of the practicals I was doing was looking at heart patients and extracting what's known as atrial trabeculae, which is it's an offcut when you do a heart bypass. And we're using that offcut with the consent of the patient, obviously, that was all done correctly, to actually look at how they interacted and what treatments they undergo. So that day would start at 5 a.m. and it would end with using a very fancy piece of kit known as a confocal laser, which basically fired at a particular frequency, and we could visualise and image the cells, and that would finish at 10:30, 11 o'clock at night. So, and that's the time length it is. That is the way that it is. You can't change that because that's how long it takes. Fortunately, I was working with a very good surgeon friend, colleague of mine, and one of us would take the early shift and she would take the late and vice versa. So we take it in shifts, sort of meet halfway between the two and then discuss it. So you're planning out your week and what experiments need to do. You normally start the week with a research meeting. We used to start the week with a research meeting at 8 a.m. on a Monday morning, which doesn't sound early, but in London, those of you who've lived in London, 8 a.m. to get to central London by 8 a.m. is pretty tricky. So you start that by presenting your research from the previous week. And every single researcher would present their research, and it would be open to scrutiny to everyone else in the group. Is it significant? What's happened here? Have you tried? And it actually was pretty brutal. And I get the impression, speaking to friends of mine who are also research scientists, that it was a pretty brutal process as well. And you get to... The risk is you get to doubt yourself quite a lot, and you're under scrutiny quite a lot, and you have to be quite resilient and push through with confidence. So someone who's starting a PhD, just as I was finishing, saying what's the most important skill or attribute you need to have as a research scientist, they expect you to say something like intelligence, but no, it's just plain resilience and pushing through. That is the biggest and most important quality.
But probably good to know early if you've made a mistake.
Absolutely.
Or if your experiment hasn't been robust.
Yeah.
I guess you find out fairly quickly if you've got a good team. Absolutely.
But because it's all brand new, you're on the cutting edge, there's no manual, most of your experiments don't work. So most of your experiments, someone said to me when I was starting my PhD, well, only 25% of your experiments work. And I was chatting to Dr. Wade this morning, she goes, yeah, that's ambitious. 25% is a good week if your experiments work. So yeah, you don't get data for quite a long time. So you're sat there in your research meetings for quite a long time with nothing to show for it. So it's pretty tough. And you just have to wait until you hit that mother load of data, and then suddenly you start moving forward. So your week starts on a Monday with the research meeting. And the idea with that is they criticise your work, but also provides a new direction about where you're going to go that week. And then you'll sit and you'll plan out your experimentation for that week. And you'll push through and you'll plan Tuesday, Wednesday, Thursday. On Thursday, we used to have a journal club, Thursday morning, where we take it in turns. One person would present all the scientific papers that have been published that week that were relevant to us, and they'd have to critically analyse and say, well, this is similar to what we're doing, we disagree with that, or this is quite interesting, they use a different concentration buffer or whatever else it is. So you have that discussion point that feeds into hopefully the research meeting of the following week. And then Friday was results day. So you do all your data and you do all your experimentation, sort of almost with your eyes closed. Oh God, I hope it's going to be all right. And it gets to Friday and you collate all your data, and then you see whether you've actually got anything that was worthwhile that week. Ready, and you try and sort of pretty it up ready for the Monday research meeting the following week. So that's a typical week really, where you get into that rhythm. And somewhere along the line in that week, I'd have a one-to-one meeting with the professor where it wasn't open to scrutiny, but it was still pretty tough business. You know, although you're in the university and you think of it's all nice and friendly, you've got to produce results. Because if you don't produce results, we can't publish papers. If we can't publish papers, we can't attract funding. Research group disappears. It is a results business. You do have to produce. So it gets pretty tough on you.
Well, but presumably, even if you do an experiment that don't work, that is useful because you know that this thing doesn't work. I don't remember the thing, but I was reading earlier, like a fake scientist making some bogus claims, and that actually somebody 30 years before had shown that was wrong. But he hadn't published the results because nobody cares about things that yield this doesn't work. So what do you have to comment?
Well, there's two things on that. The first is you're talking about serendipity. That is where you have an accidental happen chance where I make a stuff up, but actually it's an interesting stuff up that produces an interesting line. The problem is you get used to every week not producing data. Is it this variable? Have I put filters across the air vents coming into my lab? You start becoming paranoid about absolutely everything, just trying to get that data. So there's that. You do become quite paranoid if you're not getting results. That what is it this time? Because you think about we talk in year 7 about all the control variables that we talk in year 7. I got my independent variable, my dependent variable, my control variable. You still do that in research. You still have all that. It's just there's control variables you don't know about. Have I thought about all the control? Is it me breathing in the wrong place? And literally some of the scales that we're using, the balances to measure mass in the rooms, are literally affected by atmospheric pressure. So someone walking in the room at the other end of the laboratory will affect the reading on your balance. It gets pretty darn so, yeah. It's frustrating at times.
Can I ask about some of the qualities that you might get within a team of scientists, and whether you need a mixture of these qualities, or whether there are particular qualities that are valuable? So it strikes me that sometimes what I don't hear very much is the idea of being a visionary or creativity in science. Because in order to frame an experiment, someone must have had the idea that there's something in that.
Yeah.
So that's one quality that you might have. But also, do you need people that are just really good on the mechanics of setting things up, the actually making sure the team's all doing what they should be doing? What I think kind of has value?
In universities, I don't think they construct teams like that. They advertise a place by person, and they just advertise. They put out there, look, we've got funding for a PhD and what have you, or a research scientist in this, and they just choose the best person who comes along that fits that. I don't think they construct a team. But you're right. So you've got the visionary, and then you've got the person who makes things work. So the visionary is really something that we don't educate in this country. We teach them stuff, and you learn stuff about science, and you learn that if I do A and it reacted with B, you get C and D. What you don't get, as what discussed before, is the lateral thinking, where you get to make connections between, I was reading papers in geography, making connections with geography and physics and that kind of stuff, when you start having this vision where you start being able to assemble the dots. And this is one of the difficult things that you have as a research scientist, is it's all new. You can't get yourself fixated on one particular paradigm. You've got to be able to hold lots of possibilities in your head at one time. It could be A, could be version A, paradigm A, B, C, D. All of them fit the data, and I've got to hold them all equally true in my head at the same time. That is not very easy. And as you start collecting more information and more data, whether it's from a research paper or from your data, then you start narrowing down and paradigm shifting your view across to something else, to another picture. The difficulty is, of course, is confirmation bias. I've sold my papers. I've done a load of papers that says paradigm A works, therefore I'm going to want to choose data that fits that paradigm. That's quite difficult and dangerous to overcome. But yeah, so you've got the visionary, then you've got the person who makes things work. Now, when you're using kit that's at the cutting edge, I was using what's known as a flow cytometer, which measures cells, basically, and measures and labels cells. It was made by a company in Germany, and if it went wrong, they'd have to fly someone out from Germany to fix it. Wow. You can imagine my professor didn't really like that very much. So you have to become the engineer. You have to become the person who fixes it, who calibrates it. So the odd occasion where the engineer did come out, I was hovering over his shoulder. I was like, well, what are you doing there? Just so that I can learn, so we don't have to call him out next time. And he was perfectly happy with that. He didn't want to be called out to London during the week. He was quite happy not to. So you have to be able to fix all the kit and mend all the kit that you use, which is it's interesting, actually. I quite enjoyed it in the end, but it was frustrating and difficult to begin with. So yeah, making things work, and then you're helping colleagues in the lab who aren't doing. It's not a team. It's very difficult because I've got my line of research. A colleague B has got their line of research, and it will be related, but it won't be the same. I want my research to work, and if their research works as well, that's great. But I want my research to work.
So is it more competitive or more collegiate?
Neither. I think it's a bit of both. I would like my research to work more than the person next to me, but if they're having a difficulty with a particular assay, then I will help them, because it's their success will not really impact the success of my research necessarily. So you kind of a load of people who are getting on together and rubbing along, and I've got my bench and they've got their bench, and we're doing our work and we're using similar kit, and we're working with under the same professor, so therefore working in the same field, but we've all got our own lines of research. We all want our own names on the papers, basically.
Perhaps it's too gossipy a question, but have you ever had a big fallout with any of you? Has somebody ever interfered with your experiment or anything?
Interfered with my experiment? I've had fallouts with other scientists, yeah. There was one particular person that I just didn't get on with, but yeah. Interfered? No. I can't. People might be finding maybe that's why those experiments didn't work. No idea really. Not that I can think of. I think because it's you can choose to interact as much or as little as you wanted. So if you wanted to work side by side, the colleague I talked to earlier about with the surgeon, I got on very well with her, and we worked really well together, and that was a good team. And if we hadn't gotten very well, maybe we wouldn't have collaborated so much. But someone else I didn't really get on with, then you just choose your own section of lab, and other end of the lab, and you just work separately. And you can even choose your own times. So that we had core time, which we had to be in, which is sort of 10 till 2. But beyond that, as long as the research got done, I came in really early and go home at 2, or come in at 10 and you go home at 10 o'clock at night, or whatever it is. So you can choose your times, your location of the labs to be different.
I like that the Ask podcast is going a bit tabloid. Who didn't you get on
Sorry.
with? Who wrecked your experiment? This is what we need to boost listeners.
I apologise for lowering the tone.
That's a good question.
I thought it was particularly
Well,
interesting.
it's all human. I have to say, when I worked in pharmaceuticals, it is much more of a deliberate team. You will have that role within the team, and they will have that role, and you'll calibrate the machine for them. And it is much more deliberately a team to a particular end.
Could I ask about the teams? Because earlier you mentioned how research is usually individual and you focus on your own work and get it done with, but also mentioned working in teams, how you have the visionary. What would you say a typical size is for a research group?
Crikey, how long's a piece of string? I think all this is my experience, what I experienced in labs. How many people in my research group? 8 or 10 people working side by side within the same laboratory. Quite a large lab that we had. It depends. Some groups are quite small, 3 or 4. There were some people that we used to joke out. When you see their papers, they would have 20 or 30 people names on the papers, and you could almost see this university industrially trying to pump out papers. And therefore they were just throwing bodies and money and people at the research in order to be able to generate data quickly. And all those things that you see about the Watson and Crick, the race and people being the first to publish the structure of DNA. There is a little bit of that, because if you are first person, you get the higher rating paper in the higher rating journal, so you get more impact and more people read your paper. If you're the second or third person, it really is a lot less. I'll come back to Andres's point. The idea, or maybe it was Mr. Till's viewpoint, the idea that negative data, or the data that didn't work. There was an article that was in The Economist quite a long time ago, and I think may have been in New Scientist as well, that focused on the importance of people publishing data that had already been done. So if you're the first research group to publish, with the structure of DNA, what's really important is that another group checks that. Now, the problem is these days, if you're the group that is second or third checking someone else's data, it takes time and money to do that. And what you publish out of that is, yeah, they were right. And your paper you're publishing is one that nobody ever reads. So it doesn't really help you along in terms of attracting funding. But as The Economist, and I think New Scientist, I can't remember which one it was, certain point, that's a really important part of science, that someone actually checks your work. Is this actually real, or are they just making, you can make it up. The structure of DNA, quite a double helix, that looks quite nice. I draw that, that's excellent stuff. So it's quite important that people check that. And that's an important part of science, which there has been some chat about it, but I still think there's a structural problem whereby if you're the second or third, or you're the research group checking someone else's research, you don't get as high impact or as much value for that as you possibly should, and therefore it's degrading that important role.
What about the role of, so this is me trying to sound like I know what I'm talking about, what about the role of meta studies? My understanding from my brother who knows about this kind of thing Is that an area that you...
Meta-analyses, yeah. We use it a lot in teaching, simply because one of the big problems in doing any sort of research is those who do statistics for maths is we talk about n numbers. How many data points are you putting into the research, into the statistics? We use confidence, so you're trying to put in, is the effect I'm seeing real? So I pump in all my numbers into the statistics and it pumps out saying, yes, we have 95% confidence that what you're seeing is real. The bigger your n number, the bigger the number your data points you're putting in, the more confidence and the easier it is to get that confidence. Typically, we used to use a minimum number of 6. So I once worried about this and went to see a statistician at UC. I said, I'm really worried that we're not using this correct statistical package. And so I went to go and I chatted to them about what we should be doing. He goes, well, you need a n number of at least 1,000. Well, I've got 6, and I can't really do 1,000, so it wasn't very helpful. So, so what was the point? Question?
I asked about meta studies.
So meta studies. So the point of meta analyses is you can get that n number. The problem with it is you've got such a large n number. It's easier to get the confidence, statistical confidence that something is true, and so you end up with all sorts of strange things pop out of it. So you could end up with things like, yes, it is statistically significant that people who live above an altitude of 500 meters do prefer carp on their Friday dinner. Odd things. That is the slight problem with it. But I think that there's a big role for it. We never played with it much in our research, I think, because you do use it when doing clinical analyses, and I imagine they're doing lots of meta-analyses when you're doing the COVID research, because you've got those big numbers of 30,000, 60,000, 100,000. You can get a lot of data out of it. But at the level I was working at had an n number of 6. No.
At university we did a lot of discussion around meta-analysis, and we always got shouted at correlation doesn't equal causation. So
right.
I think the idea is what you're trying to look for and what you're trying to get out of them. But a lot of what we were told about is meta-analysis is helpful at sometimes, but a lot of science, although it feels quite established, it is quite in its infancy and it is quite in its baby point. So I come from psychology, of which is currently in crisis. There is a replication crisis going on, what they're calling it, of what we thought was classic and sort of empirical literature, we now can't replicate in the modern day, and sort of the ground beneath us is falling out. So I think your point on this will be really interesting, but there's a time of which to look at the past and to replicate, but there's also a time of which I think to sort of move on and to try and drive something forward.
Oh, yeah, I used to teach something called theory of knowledge. I don't know whether you know this for the IB. And it's how do you know what you know? And you can go, but why? And how do you know that? How do you know you're sat in a red chair? And those sorts of things, which really people first sit there in lower sits go, good grief, because it's red and I'm sat on it. But it's that, and then you start getting into what the stimulus and the inputs are and the causation, everything else like that. So the point we come to is you end up breaking down until you end up questioning absolutely everything, which is fine. But then you've got to be able to build it back up again and go, well, look, I just have to take it for granted I'm sat in a red chair. Yes, okay, my inputs could be wrong, and what Andres is looking at, it could be what I would view as blue. If I was plugged into his eyeballs, it would be blue. But I just have to accept that we all agree that it's red, and let's get on with ourselves, get on with our lives, So you're right. I think it's the deconstruction is necessary, provided it comes a reconstruction thereafter. And then you're holding in your head, the fact I'm sat in this chair is a hypothesis and a working hypothesis, but I'm going to work with it as though it's true.
Take a gamble almost at times.
Yeah, but I'm willing to be corrected if new data comes in and I look down again and I see that it's blue.
Can I ask quite a school-based question about A-level choices?
Yes.
So we always somewhat lament, regret, wish it were otherwise, that so many students who achieve top grades in English and perhaps in a range of subjects, they've decided that they want to pursue a scientific course, and so they go 3 science A-levels, or maybe 2 sciences and maths. Do you think there'd be an advantage, even if someone were to go on and want a scientific career, for having another subject that gives them perhaps something at A-level that is a little bit broader? I don't just mean English literature, though, spoiler alert, I do slightly, but a language as something, because out in the real world, they'll be working across countries. They might have to read papers where little language differences matter. Does someone with something a little bit different actually have an advantage over someone that's done your 2 sciences and maths, and that's what everyone else has done?
Well, I should put the qualifier in, but for the sake of Mr. Folkes, Miss Fenn, and Mr. Dunn, no, do 3 sciences.
We'll edit that bit out.
Yeah, I mean, there's a lot to be said for it. I think, it's not the fact that you're doing a language or the fact that you're doing English or the fact that you're doing music, I think, and it's the benefit I get from doing English and that the English itself will pay into a future career. I think it's the slight alteration in your mindset and thought processes that really, really matters. So, for example, we often find that those who are strong at maths are very good at their A-level sciences, and it's not because you need the maths, and because you did A-level chemistry, the maths in A-level chemistry really is not particularly complex at all.
No.
I mean, it's year 7, year 8 level, not very difficult at all. So it's not the maths itself, it's the thought process of being able to solve problems and the logic behind that. So yes, being able to do English and critically analyse things. You often find medical courses, one at King's College, which often, I shouldn't put this on the podcast, should I, selling a particular course, but they specifically look for people who are going to do intercalated degrees in something else like business management or philosophy. And I think there were some universities who quite liked people doing RS along with chemistry and biology when applying for medicine, as you might imagine. So I think it's not necessarily the knowledge you get from doing English or another language or another subject. I think it's really this slight alteration in mindset. And yeah, I think it is valuable to the world of science. If everyone did the same courses and ploughed the same furrow, you end up with very like-minded thinking. And really what you're looking for at the sharp end of the research end is lots of broad-minded thinking. People can think laterally across curricular.
Interesting.
That was a brave question, sir, because that could have really gone quite
Could have.
against us.
Could definitely have done.
It seemed to come out all right.
But no, do 3 sciences.
Oh, no. Don't undo it. Don't undo it, sir.
Doris.
That's your clue.
That's this week's clue.
So have you just given us a politician's name now? Nadine Dorris, or spelled differently?
Doris is what I will say, and
Can you spell it
you
for
can
us, sir?
put that together. Doris.
Okay, so not that Nadine Dorris then. Okay.
And so we have 2 clues on the news cycle. A large prize awaits. Do email suggestions to what the connection might be.
I've got it, sir. It's names of women. I win.
Well, we're going to have to come up with a new. Okay, no, need to be slightly more specific.
This is how good I am with clues. Not good is the short and long answer. There. Right, back to what we're doing. You really interrupted our train of thought there, sir. I feel bamboozled. I suppose, okay, fair enough. It is getting towards the end of lunchtime, and some of us, knowing no names, are teaching sixth form in 8 minutes' time. Yeah, that's me. So perhaps we better start rounding ourselves off. It's been so fascinating. I feel like I've learned a great deal, and I feel that we're in elevated company here. Hopefully these conversations will continue long after the podcast has ended. But now I'm back in. I'm still trying to get my train of thought together. I need to do thank yous. That's what I need to do, don't I? Thank you to everyone today. Thank you to our fantastic guest, Dr. Bullard, for sharing such fascinating insights into the world that he's come from, the world that he's part of, and linking that in for A-level and so on, giving us glimpses of future careers. Thank you to our brilliant panellists for your really perceptive questions and thoughts on the questions as well, adding some of your own perspectives and so on. That was brilliant. Of course, thank you to our brilliant production crew and all the work they do setting this up and figuring out what microphones. And they talk about dynamics again. Another thing I know nothing about, but it sounds wonderful.
Heroes to a man.
Yeah,
Heroes
and
to
you
a man.
can probably tell the difference in sound between when I try and edit it and when they try and edit it quite vastly. But also make sure you subscribe on YouTube. It's Jenny Summers with an I. Lovely to see lots more people coming in every time. We're on Apple, of course. We're on Amazon. We're on all the available places that you can find podcasts. And just give us a shout out to other people. I am starting to hear more people saying, I'm listening to this. I've listened to all these episodes.
Yeah.
Listen back to our back catalogue. We have a back catalogue now. So and we've got a range of different types of episodes as well. We've got a live debate from last week.
So tell your friends, tell your enemies.
Yes.
Tell your frenemies.
There's so many options.
Tell everybody.
Everybody should know. We should be famous by the end of the day. But so thank you for everything. Hopefully we will be back very soon with another episode of the Ask Podcast, and we hope you've attained plenty of secret knowledge today. Thank you very much, and au revoir.